Right.
Our genetic information is encoded by the DNA double helix.
Here is another version of that X inactivation video. I just want to show you something.
When DNA is read or transcribed, one strand of the double helix serves as a template strand. The specific order of the 4 bases encodes genetic information and serves as the blueprint for our genetic makeup. Each rung of the DNA ladder is called a base pair. In total, the human genome consists of about 3 billion base pairs and encodes about 30,000 genes. In our cells, DNA is wrapped around proteins like thread around a spool. The structure that we see here is called a nucleosome, which consists of the DNA helix tightly coiled around histone proteins. Histones have long extensions, called histone tails, which protrude from the nucleosome core. As we will see, modifications to both the histone tails and the DNA itself are indicative of whether DNA is active or not, and this is controlled by epigenetics. Epigenetics and the X chromosomes. Humans have 23 sets of chromosomes. The sex chromosomes, X and Y, determine the sex of an individual. Females have 2 X chromosomes, males have 1 X and 1 Y chromosome. Epigenetics refers to processes that instruct our cells how and when to read the DNA blueprint. As females have 2 X chromosomes.
This is what I wanted to say here. The reason I knew that this was some BS as opposed to, well, how do you know that's just not how biology works and how genes work, and that's exactly what is necessary? Because they said that it is to what dosage compensate the males for us having 2 X's. 2 X's, okay? But here's the thing. Here's the thing. How come we also have 2 of each of these genes? How come there's no dosage compensation here?
3 sets of chromosomes.
Because the X is the essence of the human. Because the X is the essence of the human. And when we have 2 active X's, we are so much more than these males could ever be. We are so much more than what we see in ourselves today. This is why. So they don't care that you have 2 dosages of each of these chromosomes. They don't care about that. They don't care about that. They only care about the one that will actually reduce us. These things are not doing anything because they are how this one, they are how the X chromosomes make the things it wants to make. The X chromosomes are the real essence of humans. That is why in the Punnett square, each individual has to have an X. And when you have 2 X's, you become a real human. You become a real human. And whatever is inherent in that, because right now we don't even know. We don't even know what that looks like. We only know what our reduced selves is. Selves are. We only know that. We don't know what we're supposed to get with 2 active chromosomes. How come they don't care about the dosage here?
X chromosomes, X and Y, determine the sex of an individual.
How come there's no compensation of women for the extra Y that they have? How come there's not?
Females have 2 X chromosomes.
Basically, with this setup, what they ended up doing was having more chromosomes, more active chromosomes than us. Do you think that might have something to do with why they are stronger? I actually think so. I don't know. Because I haven't researched that, but I'm only so far theorizing that. I think it has to do with why they are actually stronger than us. And if I'm right, then it would be quite interesting when they ridicule us for being weak.
Males have one X and one Y chromosome. Epigenetics refers to processes that instruct our cells how and when to read the DNA blueprint. As we will see, female cells use an epigenetic mechanism to inactivate or silence one of the X chromosomes in each cell to prevent abnormal development.
Did you know that one of your X's is silenced? You thought you had two X's. You thought you had two full working X's. You didn't know that one of them was silenced. You did not know that. You thought you were full human, working with everything, everything, what is that? Everything shooting off the pistols, right? You thought that. You thought everything was shooting. You thought you had two full set of guns. You didn't know the other one was what? A toy gun.
We are now looking at cells from a female body. A full copy of the genome is found in the nucleus of every cell. Each cell has the same 23 pairs of chromosomes. We can now see individual X chromosomes. Highlighted here are two X chromosomes from one cell. The inactive X is compact in appearance, whereas the active X occupies a much greater volume, despite the two chromosomes having the same gene content. We will now take a look at each X chromosome at the molecular level, which is at approximately 1 million times magnification. The active X chromosome. The active chromosome has a dispersed or open appearance, often described as resembling beads on a string. Molecules involved in gene transcription can access open DNA. The active X chromosome has specific active histone tail modifications that tag the DNA. In the next few shots, we'll look at examples of molecules that can access the active X chromosome. Transcription factors can bind open DNA. These proteins are important in transcription initiation. They can recruit other proteins, such as histone-modifying enzymes. A new modification is added to a histone tail. Open DNA is more loosely bound to histones, which allows for nucleosome disassembly. Nucleosome remodelers can also access the active X chromosome. These proteins slide the nucleosome along DNA. Ultimately, these changes open the DNA and allow RNA polymerase, the DNA-reading enzyme, to access and transcribe DNA into messenger RNA. The inactive X chromosome. The inactive X chromosome is more tightly bound and condensed than the active form. This means that the DNA is less accessible to molecules involved in transcription. Inactive X has modifications, including specific inactive histone tail modifications, DNA methylation, and additional structure.
Actually, I don't think I was saying the right things earlier, and I kind of want to go back and correct that. Okay, hold on a second. Okay, here what I was saying was that if two dosages are okay here, why would two dosages be not okay when it comes to women? Do you understand that? Why is it okay for you to have what this one said was they inactivated because the woman would be weird or something to that extent.
DNA review. Our genetic information is encoded by the DNA double helix. When DNA is read or transcribed, one strand of the double helix serves as a template strand. DNA is wrapped around proteins like thread around a spool. The structure is simple. Humans have 23 sets of chromosomes. The sex chromosomes, X and Y, determine the sex of an individual. Females have 2 X chromosomes, males have 1 X and 1 Y chromosome. Epigenetics refers to processes that instruct our cells how and when to read the DNA blueprint. As we will see, female cells use an epigenetic mechanism to inactivate or silence one of the X chromosomes in each cell to prevent abnormal development.
Normal development. To prevent abnormal development. This is BS. This is the stuff that they come up with to explain the things that they are seeing, which are the things that were put in place to enable their existence. So what they're actually doing, what he's actually doing, is explaining how he is important to the entire process. Where if you can explain this any other way, it would mean he has no importance to the process at all. That he should not even
Highlighted here
be
are two.
here. That males are literally worthless. This is why they have to come up with all this nonsense to describe it. Abnormal? Abnormal? How come? How come? How come abnormal doesn't occur with these? What's this thing? I've got to find it. What's this thing? How come abnormal doesn't occur with one? With the first set of two chromosomes, two pairs of chromosomes. How come? How come? He's going to show a calico cat. He's going to show a calico cat. Right? This calico cat has this because it has its X inactivated. I explained it in the other one, but actually no, they explained it and they explained it here too. So let's go over that.
While it is not generally visible to the naked eye in humans, you are probably familiar with this pattern as we see it regularly in calico cats. In cats, the gene that encodes coat color is found on the X chromosome. As a result, only female cats can inherit two different coat color genes. If this happens, she will exhibit that characteristic tortoiseshell coat because of X inactivation. Why is epigenetics important? The epigenetic regulation of DNA is not peculiar to the X chromosome.
The reason,
as a result,
I don't think it's going to take fun. So the reason it happens, a calico cat happens because of X inactivation is because in the X inactivation, the embryo comes from one singular cell, and then that cell divides into hundreds of cells, right? And then that's when the X inactivation happens. And the X inactivation happens by silencing one of the X's of every single cell of those hundreds of cells in the blastocyst stage of the embryo developing, okay? Of the embryo developing. And this happens to basically every species of animal that has this problem. Yeah? This happens in them. So the embryo at that stage, they do it to hundreds of cells. Because if you wanted it to be inherited by every single cell, you will have had to have done it to the progenitor cell, to the singular progenitor cell, which will be the egg. Yeah. So you would have had to do it at that stage. But because they did it at a later stage, which when it had more than one cell, right? That's why it has all of this. And the X inactivation is turning off one X in every single cell, right? So even though the cat has two X's, they will turn off one X so that it only has one X to work on, one X to live on, one X to drive its biological processes. And because there is an X from a father and an X from a mother, and they have the different, and they would have different color coat, color fur on each of them, right? So whichever embryo has one that has one color and the one has another color, right? That's when they would have this issue, this calico, this calico cat thing. Now, they were saying that they don't want the female to develop abnormally. Isn't this abnormal? Isn't this calico cat with these two different colors, color skin, color fur, isn't that abnormal? He showed a human with two different colors. Isn't this abnormal? They don't want you to develop abnormally. Isn't this abnormal? What I'm telling you is that the excuse of abnormal, that is the lie. That is a lie. They turned it off so that we cannot do parthenogenesis. This is the source of, well, this is one of the source of the parthenogenesis being blocked. It's not dead. It's not non-existent. It was blocked. It was blocked. So they're telling you all these things. Oh, the female will be abnormal, but here's abnormal skin. Who wants skin like this? That might not care that it has those color, different colors first. But we as humans, we do. We care that we have two different color skins like this, right? And they tell you that it's so the female is not abnormal, but you have that issue, which means that's just a lie. That disproves that, because it does in fact create abnormality. And then there's also this, all these other issues you have. How should I say? You have eye color. Eye color could be on one of these. I don't know. I actually don't know that. But there are many other things that could be on all these other chromosomes, right? Doesn't having two of them create a problem? Doesn't having two of them active create a problem? What about the men? What about the men? Is there any abnormalities in them for having two chromosomes of each one? Two set of each one? Again, why is it only the X? This is the lie that they tell you. They tell you they're doing it to fix to fix the woman so she's not abnormal. That's the lie. That's the lie. They're fixing you so that you can do parthenogenesis. And then in the previous video that I did, there was, I showed how they're doing it. They're doing it to the embryo, which means that it is not, God, what is this word? It is not heritable. It is, oh God, what is this damn word? It is what's it called? Non-hereditary, yes. It is non-hereditary. And it is not, let me explain to you why it's non-hereditary. Oh my goodness. The mere presence of two X's creates this parthenogenesis. The mere presence of it. That is why when they block it in your mother, it doesn't transfer to you. And they have to run this process on every female embryo. The mere presence of two X's is what causes this. It's what creates parthenogenesis, because that is a full human. You are like a plant. You see how a plant, you just, in order for you to have two plants, right? You can just cut off some parts of the vegetative part of the plant, and you have two. That is exactly like that. It can propagate because every part of it is on parthenogenesis. That's exactly what that is. Just a simple presence of two X's is what causes you to be parthenogenic. The simple presence of two X's is what causes you to be parthenogenic. And that is why they are silencing the other one. That is why they are silencing the other one. Because by it being silenced, you can no longer parthenogenize. And now you need what? A male.
Modifications that tag the DNA and allow RNA polymerase, the DNA reading enzyme, to access and transcribe DNA into messenger RNA. The inactive X chromosome. The inactive X chromosome is more tightly bound and condensed than the active form. This means that the DNA is less accessible to molecules involved in the enzyme to access active X chromosome. The inactive X chromosome enzyme to access and transcribe DNA. This means that the DNA is less accessible to molecules involved in transcription. Inactive X has modifications, including specific inactive histone tail modifications, DNA methylation, and additional structural proteins that help to zip up the DNA. Ultimately, this
part of the work that we are going to do, we are going to actually get into a lab, is to research how to undo this, okay? And I've actually already gotten started on that. And the way to undo this is by us. You're not gonna understand it. It's by acetylating this, whatever it is that is holding them together, which is the methyl groups. It is the methyl groups that is causing them to behave like this. And so once we can acetylize it, right, it then becomes a stable form, a less stable form of itself, and then it kind of just falls apart, and then we can just unravel it, right? So part of the work that we're going to be doing is going to test this on some animals to see if we can re-enable their parthenogenesis. Because remember, it is inherent in them. It is inherent in them.
Version of the chromosome is unable to be transcribed and is therefore silenced. So let's compare the two X chromosomes directly. The active X has a dispersed or open appearance. This means the genes on this chromosome are active or on. The inactive X has a condensed or closed appearance, meaning that the genes on this chromosome are inactive or off. Histone tail modifications and DNA methylation are examples of epigenetic marks, modifications that distinguish between the active and inactive chromosomes. Epigenetic inheritance can be visualized by the process of X inactivation. This is a close-up of a single cell from a 100-cell embryo.
When he says inheritance, it means inheritance by the cells of that one female that this was done to, right? Not by the actual baby of that same female, right?
Embryo, which is approximately 4 days old. At this early stage of development, female embryos still have two active X chromosomes. One X chromosome is inherited from the father, the other is inherited from the mother. Around this time, the two active X chromosomes interact transiently and one is inactivated. The other is kept active. This process is mediated by both RNAs and proteins. The inactivation of the X chromosome involves the condensing of DNA. This is marked by the change in histone tail modification, the arrival of structural proteins which help condense the DNA, and DNA methylation. X inactivation is complete at the end of the 100-cell stage of embryo development. The decision of which chromosome to inactivate is surprisingly random, and individual cells can have either the maternal or paternal X chromosome remaining active. However, from this point on, each cell will remember which X chromosome it has activated and pass this information on to its progeny as the embryo develops. This is an example of epigenetic inheritance. The same pattern is maintained into adulthood. Let's look at the female adult. If we could visualize X inactivation on the skin, you would see a distinct pattern based on the growth and migration of the original 100 cells in the 4-day-old embryo. While it is not generally visible to the naked eye in humans, you are probably familiar with this pattern as we see it regularly in calico cats. In cats, the gene that encodes coat color is found on the X chromosome. As a result, only female cats can inherit two different coat color genes. If this happens, she will exhibit that characteristic tortoiseshell coat because of X inactivation. Why is epigenetics important? The epigenetic regulation of DNA is not peculiar to the X chromosome. In fact, it is used throughout the body to turn different genes on or off in different cell types. For example, the insulin gene is...
It actually is very peculiar to the X chromosome, and here's why. He started talking about, how it is used to control genes, to turn genes off and on, etc. Yes, genes. This would make perfect sense for genes. This makes perfect sense for genes, for you to turn off an entire chromosome. That makes no sense. They turned off an entire chromosome, and it's not just any chromosome. It is the exact one that you would need to be a whole human. That is definitely doing something significant. That is doing something that is noteworthy. And the way they keep explaining it as to make it seem so reasonable. They cobbled us. Not being able to parthenogize is literally like killing the plant.
Switched on in pancreatic cells where insulin is made, but is inactive elsewhere in the body. Antibody genes are active in the antibody-secreting plasma cells of the immune system, but switched off elsewhere.
Are you noticing? Are you noticing what they're saying? Genes versus chromosomes. Genes versus chromosomes.
In the body. And just as epigenetics is important for normal cell function, when things go wrong, it can lead to disease. For example, there are certain genes in our bodies that
Thank you. Thank you.
Cancer.
This is when you know something actually is not correct, when it does in fact lead to disease, because the disease state is not the norm for any organism or any genes.
Collectively called tumor suppressor genes. These genes are usually on. Some diseases can cause the aberrant inactivation of these suppressor genes, which can lead to cancer. Scientists are trying to understand how inactivation of these suppressor genes, which can lead to cancer.
So tumor suppressor genes, that means our body has a thing where it doesn't, I mean, like you probably heard cancer suppressor genes, right? But what it actually is doing is a system to control genes from going amok, right? So the absence of that is what then turns genes to go amok. Now you need to understand this. If you as a if a human being is if a full human being is 2xx, and that is evident by the fact that you should be able to parthenogize. That is still a theory right now, right? We'll have to get into the lab to prove that. But this is a theory that I am 1000% behind, that I'm 1000% sure of. Okay, so if you're being able to parthenogize makes one human, because that is inherent. Being able to reproduce is inherent, is an inherent part of a cell. Therefore, it is also an inherent part of an organism. Yeah. Okay. And now the males have one. The males have one chromosome, which and because they only have one, they cannot procreate, right? So what does that tell you? That they are abnormal. They're not being able to procreate means that they are in fact abnormal. So what does that tell you about the cancers that we are seeing in society? And they have done that to us in a sneaky way. They have done the same thing to us, where even though it looks like we are perfectly functioning humans, we are actually not, because half our genes, half our X genes, half our essence are missing. Do you understand why the cancer is happening now?
Cancer. Scientists are trying to understand how epigenetic regulation controls normal development and how epigenetic mistakes cause disease. By understanding the mechanisms involved in epigenetic control, they will also start to look for new treatments that will override the epigenetic marks that cause disease.
Hey guys, welcome back to Now I Know. In the last video, we were talking about X chromosome inactivation. We had seen the overall process of lyonization, bar body formation. We talked about dosage compensation, and we saw the effect of X inactivation on the phenotype in calico cats.
Actually, it for now. Next time, I'm also going to talk about the diseases that are caused by this very X inactivation thing, right? Because they're saying, because they said that this was done in order so that women are not abnormal, but what ended up causing is diseases. So which is it? The absence of it is abnormal, or the presence of it, of this X inactivation thing, is what causes diseases? Which one is it? Because it can't be both. It can't be both. Both options cause diseases. One of them has to not. One of them has to not. And of course, the proof that it's not being activated, or it's being activated, like having a two full cells is the one that doesn't cause diseases, is the fact that not having two full ones is what causes diseases. That is your proof. That is your proof.